Hey, hello and welcome everyone. So today we're going to be talking about mixed method research design. And I'll start by saying there's two interpretations of what this means. We'll start with the first one, which is the topic your book covers, which is factorial mixed designs. So what is a mixed research design or a mixed factorial research design? This is a approach that uses both within subject and between subject independent variables. And it's a very common type of factorial design, which we talked about a couple of weeks ago. And in this particular aspect, all participants are exposed to one level, that's that within subjects, and only some are exposed to that other level of between subject. And it's a very commonly used approach, especially in behavioral sciences and psychology, because it's better able to test some of the complexity of effects that are combined over time. So an example here would be learning over time by various group conditions. And I also have an example down here of if I were to be studying the effect of therapy and different types of therapy, maybe I'm looking at cognitive behavioral therapy versus mindfulness, and I want to look at that both within between subjects and test for time, so pre, post, follow up within groups and between groups, I would be able to do something like that while still looking at its effects specifically on anxiety. So I might be grouping people based on their baseline of anxiety. Do they already have high anxiety? Do they have low? How does that test given the different conditions? And then how does that change over time? So things get much more complex when we're studying people, which is why these factorial mixed methods research designs are increasingly popular. And of course, there are lots of different ways to do this. So let's walk through this table, which is something I borrowed from your textbook, and talk about the most common type of experimental designs and whether they use that within subject, between subject, or mixed methodology. So starting here at the top, and a lot of this is a recap, just tying it all together, we have the two group design, which is a very simple between subjects setup. So this is where we randomly assign participants to one of only two groups, and each group experiences a different level of that independent variable. So often this is a control group versus a treatment group. Simple, two. The pre-post test design is within subjects, and this is where we're having the same participants are being measured before and after some sort of intervention. And since all participants are go through both stages, it's considered that within subject design type. We then have a multi-group design, and this is also between subjects. This is when, kind of like the two-group design, but with more than two levels. So here we might have 3, 4, or more conditions that all participants are assigned to only one level of. Then we have that factorial design, which in its most traditional form uses only between subject variables. So if we have two independent variables like the treatment type and the setting of that treatment, you might assign different groups to each combination of those things to test for differences between it. We also have a repeated measures design, and this is within subject. This is where all participants experience all levels of the independent variable, and it's very efficient in terms of like sample size, so you can use less participants and it increases that statistical power, but there are more risks of those carryover effects we talked about a few weeks ago. And then it leads us to what we're talking about mostly today, which is that mixed design, which combines multiple of these approaches. So some of the independent variables are tested between groups, and some of them are tested within the same group. So it's really helpful, again, especially in behavioral sciences and psychology, where we want to compare those groups, but also track changes over time or under other conditions. So we can focus a little bit more on mixed methods today, of course, but I find it's helpful to give a recap of kind of what we've been covering and how it then informs this mixed design. So what are some of our pros and some of our cons? So our pros is that when you're combining different types within and between, you get the strengths of both. So we get that internal validity increase from using between subjects, and we also have a stronger statistical power from within subjects. It allows for more nuanced and hypothesis testing. So we can test for a lot of different interactions. Of course, it does, well, I'll talk about cons in a second. But yeah, it lets us get at more complex research questions, which is very common, especially within our field. It's very rare that we're able to isolate everything. And so oftentimes, by the time we find eligible participants, we want to make sure that we're getting a lot of information out of that study. And it's very efficient. So it's fewer participants than if we were to do a strictly within subject part or within subject testing design. But of course, there are complexities and cons. The con being the complexity a lot of the time in both design and analysis. So you have to think even more about carryover and order effects. So what order are people being exposed to your different stages, your different variables, your different settings? It usually is very helpful. This is true in general, but it's especially true in any sort of mixed factorial design. You really want several people on the research team just because there is always another layer to be considered. And they take a lot more time to design for that purpose. It just takes a lot more intentionality. And part of it is because while you are getting both the pros of these different approaches, you're also getting both of the cons of those approaches. So there's more threats to validity when you're looking at both of those methods. So again, those carryover and selection effects. It can be more challenging to interpret the findings as well and the interaction between variables. But again, a lot of that is stuff you control for in the research design phase. So you should know what statistical approaches you're going to use well before you actually collect that data. But honestly, the biggest con is just that it's a lot harder to design and it requires a lot more intentionality. So there we go. Pros and cons, as there is to, it turns out, everything in life. So this is another table that I am borrowing from your textbook because I like how it summarizes a lot of things. And this is really to help us better understand what makes mixed design so powerful, but also more complex, and revisit some of those differences. So let's start with the method used, and I'm looking over here. So in a between subject designs, each participant, again, is only exposed to that one level of independent variable. And a drug trial is a very classic example. One group gets the medication, the other group gets the placebo. And that's in contrast with that within subjects, which is where all participants are exposed to all levels of that independent variable or treatment. And using that same example, a participant might try both the medication and the placebo at different times. They don't know which is which, though. Excuse me. Next, we get into the component of random assignment. So in between subjects, participants are randomly assigned to either of those single conditions, whereas within subjects, they're randomly assigned to a sequence of treatment conditions. So it's not just their group A or group B, it's that the pattern that they are exposed to will also be randomly assigned. So do they get the placebo first? Or do they get the medication first? All of those components also have to be randomly assigned. It's not enough to just put them in groups. You have to think through each individual participant and how to randomize that so that you can minimize for things like carryover effects. Some of the strengths is that within our between subjects, we do have those stronger aptitude for internal and external validity, especially when you are relying on that random assignment. It's a lot easier to do when it's smaller assignments you're having to make. And then within subjects, you get a lot more in terms of statistical power within subject design, since participants are serving as their own control group, and it reduces variability to some of the individual differences people may have. So if I'm just using a between subject and some of them get placebo and some of them get a medication, there's going to be natural and normal variances between the participants. No matter how much I try to plan to make sure that everything is even, there's going to be some participants that are higher on the whole collectively of maybe something like anxiety or whatever. But if you're using the same participants as the the comparison point, like you do within subjects, it controls for a lot of those variances quite a bit more, because I'm able to say more confidently, okay, it's not the difference between participant groups because it is the same participant. It has to be then something about the order of treatment or about the treatment itself, depending on your research question. So, and again, this is recapping just the weaknesses between subjects. You have less power in terms of that statistical understanding. And within subjects, you just have a whole lot more threat to that internal and external validity. So fatigue, practice effects, and participants started to like guess what it is that your independent variable is. So I know I talked more about that, but this is really just a recap as we're pulling everything together throughout the semester. Okay, so some big components of control groups within these mixed factorial designs. So first we have a treatment as usual approach, which is where participants are receiving standard care or best practices, and it's used to test if a new treatment will outperform their real world performance. So an example of something like this might be if I am looking at the efficacy of cognitive behavioral therapy versus just medication versus treatment as usual, which is any type of treatment that they're finding out in the wild, I would have those groups. I would have a controlled group where they're all getting the same type of cognitive behavioral. I would get a controlled group where they're just getting medication. And then I would have a controlled group where they're having treatment as usual and I'm not messing with anything. There's going to be a lot of variance, but that is still average, normal human experience. So control probably was a confusing word to use there because it's not a control group. That's not what I mean. I just mean as an experimenter, I'm able to control or manipulate those components more, but I see where that might be confusing. So let me add that asterisk. The treatment as usual is often helpful in a between subject design or these mixed designs that are including a component of between subject. And we just talked about some of those examples. And that is contrasted with something like a waitlist control group. And this is where participants receive no immediate treatment, but they are promised it later. So rather than saying, go about your usual form of treatment for this issue, we're saying, no, we don't want you having any treatment at all for this issue, but we will give it to you later. It is usually later, is typically after the study is concluded. And for ethics reasons, we can't say that some participants never get access to the treatment, some of them do, but sometimes we need that for the science comparative purposes. So we will instead waitlist a control group, and then after we're done collecting data, we'll also give them the treatment if they still want to do that. So it's used to test that treatment versus no treatment baseline a lot of the time, and it is very common in between subjects and again in these mixed factor designs. And we're able to then compare them across time within each group. So really the two big differences here is that both of them are kind of serving as a control group in different ways, but treatment as usual, we genuinely don't manipulate anything, which means there's going to be a variety of how people are managing whatever symptom or variable we're interested in. Whereas the waitlist control group, we are saying, no, we all want a standard approach of no care for this thing, but later we'll treat you. And the role in mixed methods, again, is that it lets us allow for those group comparisons, and it's very helpful when we're combining it with a time-based component of measurement. So if we're, and I'm picking on treatment here just because it's helpful to talk through examples, it's really ideal to use some of these approaches when we're evaluating something like treatment. And, if I had a magic wand and can do it all, I'd probably have a little bit of both. I would have a treatment as usual control group. I would have a waitlist control group, and I would have my different levels of treatment. So I can say, okay, so here's what happens when they have no treatment in that waitlist control. Here's what's happening when I'm not messing with anything. This is just how they normally treat it. That's my treatment as usual. And then I can manipulate all of the other components that I want to and test for those effects, especially over time. So both of them can be a type of, or be a part of a mixed factorial research design. Let's see, and this is really just trying to recap some of this information that I think can be helpful. Don't need to repeat it just for the sake of it, but I like to throw things in a table because, at least for me, it helps me collect lots of aggregate information in a way that's helpful when I am referring back to it. So, okay, we're using a factorial mixed methods design. What does that mean in terms of statistics? And per usual, the type of statistics you're going to use is really dependent on your design structure. So again, some of this is a recap, putting it all together. But when we're using a just between subjects, you're probably going to use something like an independent samples t-test, where we are comparing the means between two groups, or a one-way ANOVA. Within subjects, we are probably going to use a paired sample t-test, which means we're looking for differences within the same group of people over time, or repeated measures ANOVA. And for mixed design, we're usually going to use something called a mixed design ANOVA. This is also sometimes called a split plot ANOVA, but the textbook and I like to call it a mixed design ANOVA. And part of what you're going to consider as you're picking your statistical approach is how many independent variables you have, how many measurements you're having, if you're collecting those over time, and whether the participants all receive the same conditions and treatments or not. And then we'll hone in a little bit on this mixed design analysis of variance, which is what ANOVA stands for. So mixed design ANOVA, mixed design analysis of variance. And this is when we are studying something that has both those within and between subject variables. And there's three main effect types that you are testing for. So the first is that test between subjects, like we talked about. The second is the test within subjects. And the last one is the interaction between both the within and between both the between and the within. So a lot of the same words used over again, but that's okay. And a combination of this, what literally mixed design ANOVA is a combo of repeated measures and factorial ANOVA. So we're doing both in that way. This also produces an F statistic, which, as a reminder, has the F statistic is basically the difference relative to noise. So how sure are we that we are testing the effects that we think we're testing? And the p-value is telling us whether that F is statistically significant or not. So that is whether the differences we're seeing are due to manipulation versus random chance. And in psychology and behavioral sciences more broadly, we're usually looking for a cutoff of 0.05 for something to be considered statistically significant. So that's that p-value of 0.05 to say the result is statistically significant. A lot of this is a recap. That's just the nature of it, but hopefully this is helpful. And there's another way we use this very similar term. So instead of mixed factorial methods design, which is what your textbook primarily talks about, there's also something called just mixed methods design, and that's a little bit different. So in just straight up mixed methods research design, this is referring to a very wide array of design approaches where we're combining both quantitative and qualitative data to make sure we're getting the same picture. So quantitative, as a reminder, are any sort of numbers or statistics. So we are trying to turn experiences into numbers. So on a scale of 1 to 10, how anxious are you? Does that change over time? Qualitative is words. So this is more descriptive, it's thematic. I'm not trying to quantify something. It is me collecting stories and then looking for themes in those stories. Quant and qual. The way I've talked about it so far is that a research design is either quantitative or qualitative, but it is increasingly popular, especially in behavioral sciences, to use a little bit of both and make sure that they're measuring the same thing. So if I ask 100 people on some sort of Likert scale about their anxiety and how that has changed as a result of the treatment, I'll get a certain picture of how statistically significant that is, and I'll make decisions based on that. But sometimes if I ask those same group of people to just sit down and talk to me for 15 minutes and I ask them, what about the treatment do you think is helpful? Sometimes it is the treatment. And then I'm like, great, because that means that my statistics align with your story. Boom, we can be even more confident those statistics are correct. But sometimes what happens is you sit down and you talk to people and you say, okay, what did you like about this treatment? And it's not the treatment, they like the therapist. So that's really important information I didn't collect in my little Likert scale about their anxiety. There's this other factor that maybe several participants say, yeah, the treatment was cool, but honestly, I really liked my therapist. The therapist was wonderful. And it turns out the therapist was the only one who was involved in the study. So it's actually the therapist and not the treatment itself. And I wouldn't have known that if I didn't sit down and collect some qualitative information. So especially in big research experiments or things that are well funded, nowadays you'll usually see a little bit of both. I argue that it is a stronger research design, especially in behavioral sciences, because it helps us make sure that we are interpreting the numbers correctly and assigning the correct understanding of what it is that made that change over time, that we're not missing some of those confounding variables. In my example, it would be something like the therapist themselves who was delivering the treatment rather than the treatment. So I think I already covered why. So with more complete understanding, it validates that finding through triangulation. Sometimes there's a fancy way of saying we're making sure we're getting the same story. Triangulation is how we discuss that. And it also explores not just what happens, so these things are different, here is the outcome. It's also the why and the how, and more of that like phenomenological experience that someone might be having during those treatment conditions. We talked about my example. This YouTube video I really like as a review of mixed method research design. And what I mean by mixed methods is that quant and qual combination. There are three major types of mixed methods that we often talk about. There are more than that, but these are the three big hitters. The first one is called explanatory sequential design, and this is where you collect that quantitative numbers data first, and then you follow up with qualitative. So an example would be a survey showing low test scores quantitatively, but then I follow up in some interviews and try to understand why the students think that's happening. So, okay, I found this interesting thing, this interesting result in my quant. I don't quite understand it. Let me follow up in this explanation, sequential design to understand and contextualize some of those findings. I will say a lot of the time this research design is not planned out. It probably should be, but a lot of the times if there's a big research project going on and they get all these statistics and they don't make sense at all, the next step will be to pivot and move towards this mixed design. So you have this sort of extra explanation to help you understand the numbers and the outcome. We also have an exploratory sequential design, and this one is more like required that you replan it, which is good probably. And this is where we start with qualitative first. So I might start with those interviews and just asking people, what is this experience like for you? And then I use that information to inform how I ask them quantitative questions. So I've done a lot of work in measurement development of like, how are we developing this quantitative measure to assess for something like anxiety? How are we sure it's actually asking questions the right way? How are we sure that people are understanding the questions the way we mean them to? Something called cognitive interviews. And that's what we did. We started with a lot of qualitative interviews. What is this experience like for people? Once we start getting themes, then those themes will turn into individual questions on Likert scales. And then we will quantify those experiences so we can test in bigger groups of people for differences between them. So really helpful when you're developing theories, when you're developing surveys or different types of interventions. So focus groups are a very common format of this, very common in business as well to have those focus groups. And then once I understand what that experience is like, I might turn it into a scale, and a scale is a lot easier to collect information from a whole lot of people, whereas it takes a lot more time to talk to people individually and genuinely listen to them. And then we have something called a convergent parallel design. And this is when we collect both quant and qual at the same time. It's not that same temporal, like quant then qual, qual then quant. We collect them both at the same time from the same group of people. We analyze them separately, and then we merge them to make sure we're getting the same information from both and make sure we're not missing anything in either of those approaches. So we compare and contrast to make sure we have that really comprehensive understanding. Yeah, okay. So this is our last slide of this particular lesson. No, it's not one of our longer ones, but it is a recap. And this is talking about each of our design types I just covered and sort of recapping the sequence, purpose, and the pros and cons of each of them. So as you're thinking through your assignments for the week, this will probably be helpful to answering some of those questions. So really just a recap. But again, the two big takeaways is that mixed research design actually kind of means two things. We have that mixed factorial research design, which is combining within and between groups. And we also have this mixed research design, which is both qualitative and quantitative. I wish that they had distinctly different labels for them, but they don't. So that can be kind of confusing sometimes, but that's okay. So tying it all together, things get more and more complex, but as always, please reach out with any questions you may have, and I'll see you guys next week.